Hi, I'm Jay Phelan. Let's talk about some biology. Today we're going to talk about something related to cell division, which we've been discussing, but it's the downside. It's what can go wrong in a process that's this complicated. So in order for cells to divide, a huge number of events need to happen that involve the cytoskeleton being built up, changing its shape, pulling things, moving. And that doesn't always work just right. And if it goes wrong, a common thing that will happen is that when you're supposed to have equal proportions of genetic material pulled to the left side and to the right side, you can end up with too much getting pulled to one side, not enough getting pulled to the other side, and that can lead to problems. It can occur in three different points during cell division. It can occur in meiosis, the production of gametes during meiosis 1 or during meiosis 2. Meiosis 1 separating the homologs, meiosis 2 separating the sister chromatids, and it can occur during mitosis. Because we're interested in the heritability aspect of genetics here, we're going to focus on the meiotic cases of non-disjunction, which can lead to problems that we see in offspring that are born. We're going to look at meiosis 1 in an example here. So as we're proceeding through the process, metaphase 1 of meiosis is defined by the fact that the homologous pairs all move together to the middle of the cell. We call it the metaphase plate, and there will be the sister chromatids of the maternal homolog and the sister chromatids of the paternal homolog together in a tetrad for chromosome number 1, number 2, number 3, number 4, and so on. All 23 pairs in humans. In the drawing here, I've only shown two, a blue one and a green one, with, we could say the darker one is the maternal, the lighter one is the paternal, and that's what they would do. So now typically in meiosis 1, we'd be pulling them apart, not separating centromeres or anything, but just separating the homologs. But what could happen is with some connection of the cytoskeleton and some pulling, for the green one here, I've shown both of the homologs are getting pulled to the left side. That's going to leave too many on one side. It's going to leave too few on the opposite side. And then what happens is when that division is completed, now we're not done with meiosis, but for this first division now, we've got three sets of sister chromatids on one side. We've got only one set on the other side. So we can see that things are going to go wrong. We're not going to end up with gametes that have one copy of every allele, one copy of every chromosome in the cell. So at the top here, we have the same situation I had from the previous image. But now, in meiosis 2, we separate the sister chromatids. So this is where the duplicated identical versions that aren't identical because crossing over has occurred, the centromere splits, and they get pulled apart. So typically, if we had had an even division the first time, then we separate the sister chromatids here, every cell would have the same amount of genetic material. But instead here, since we had three homologs on the left side, now when we separate the sister chromatids, we get two different gametes that each have three, and then on the other side we have two gametes that each have only one. So the resulting gametes that come about, whether this occurs in the female and their eggs, or if it occurs in the male and they are sperm, they're going to be messed up. So you see, for instance, on the left side, there are two green ones. There is the dark green, the maternal, and the light green, the paternal. So you'd have two copies of that allele. And think about that. That's an egg. Later on, when it gets fertilized by a sperm, you're going to add in a third copy of an allele. And that's not going to work out. I showed there how it could happen in meiosis 1, that the homologs don't get split evenly. During meiosis 2, though, we could have the same problem. You split, all the homologs go perfectly to the left and right side, but now you've got these pairs of sister chromatids. So in the second division, where you separate those and one goes to each side, same thing could happen. Whoa, two of them go over to this side, then none go to this side for one of the chromosomes. If that happens, you end up with the same thing. You have a gamete that's going to have an extra copy of that chromosome, and you have another gamete that's going to have nothing. So you end up with the same problem, but every time genetic material is supposed to be split evenly and go to either side, you can have non-disjunction occur. Here's a picture during anaphase 2 of meiosis. We know it's anaphase because we've already seen it split into two, but then we're splitting again into the second one. You get a sense of all the stuff that has to be pulled from one side to the next, and stuff gets tangled up. Cytoskeleton doesn't attach properly. There are a lot of reasons why it can not divide evenly. It seems amazing to me that it divides evenly ever. So our take-home message is we define non-disjunction as an unequal division of the chromosomes during cell division, and we want to keep in mind that it can occur when the homologs fail to separate in meiosis 1 or when the sister chromatids fail to separate properly in meiosis 2. But in either of those cases, it leads to a gamete that has the wrong number of chromosomes. It's going to have either 0 copies of one of the chromosomes or 2 copies of one of the chromosomes, and then at fertilization, that's going to be problematic. We can detect that non-disjunction has occurred in the production of an organism if we look at a karyotype. Karyotype is an organized display of all the genetic material from a cell in an organism. Here we see 2 copies of chromosome number 1 and number 2 and number 3 and so on. The way you would get a karyotype typically is through amniocentesis. So you'll have one person will operate an ultrasound to detect where the fetus is so that you can get cells. You're trying to get cells that have sloughed off of the fetus so they have the fetus's DNA so you can look at it. You'll take out some fluid. The doctor does it by taking this long syringe, no anesthesia. They jam it straight through, and then they guide it with the ultrasound. They'll pull out the fluid with the cells. And typically in the cell, they're going to look like this. They're not color-coded. They're not organized neatly. But someone who's an expert in this, they can just look at them and say, okay, there, that big one, chromosome number 1, that's fine. Chromosome number 2 pair, that looks fine. But they might see something like this. In this case, if you were an expert, you'd say, ooh, there's a copy of chromosome number 21. There's the other one. And this is from a developing fetus, so it should be diploid, should have 2 copies of everything. But then they say, there's a 3rd copy of chromosome number 21, meaning that somehow during fertilization, either the egg carried 2 copies of chromosome 21 or the sperm did. When they came together, you end up with 3 copies. We call it trisomy 21. Typically, they'll do this on a computer monitor. They'll have the cells video camera attached, and then they literally just cut and paste things until they organize everything in its proper order. When you set up the karyotype then for an individual where non-disjunction occurred and you get a trisomy 21, it would look like this. So you just have, instead of 46 chromosomes, you have 47. Now, we hear about trisomy 21 and Down syndrome pretty frequently, but we see babies born with trisomy 18 or trisomy 22 and several others. But we never see trisomy 1. We never see trisomy 2. We never see trisomy 3 or 4. What's going on there? Why do you think that is? There's a reason. I'm going to let you think about that one. Here's a curve that maybe you've seen or you've at least heard about, which is the incidence of Down syndrome births to women of increasing ages. And you see that as the mother is older and older and older, the incidence of Down syndrome goes up significantly. Beginning around 35, it starts to get pretty steep until at 45, it might be, 5, 6, 7 times higher than it would have been at age 30. So the question is, why is this? Why are babies born to older women more likely to have Down syndrome than babies born to younger women? Now think about this, how the mother is producing her eggs. Meiosis starts in the production of eggs very early in her life. But it doesn't complete until she's going to ovulate that egg, so much, much later in life. So think about maybe what could have happened there. Trisomy 21, though, is one way that an individual can be born, and there are some health issues, but the individual can live. Life is also possible if an individual is born with too many or too few sex chromosomes. So the X and Y, the 23rd pair, if you're female, you have 2 copies of the X. If you're male, you have 1 X and 1 Y. But in the production of sperm and eggs, you might end up with an egg cell that has 2 X's because of non-disjunction, or no X's. Or you might end up with a sperm that has 2 X's or no X's, or 2 Y's. And when one of those gametes is involved in fertilization, then we end up with certain types of outcomes that happen, not frequently, but the individuals can live. So, for instance, in the first one here, we have one X chromosome and then nothing in the other slot. So the individual only has 45 chromosomes. We call it Turner syndrome. And it can come about in two different ways. You have a sperm with an X chromosome and an egg that didn't get a sex chromosome. Or you have an egg with an X chromosome, they're all going to have those typically, and a sperm then didn't get one because of non-disjunction. Or we have something called Klinefelter syndrome. This is where you have two X's and a Y. Now, how could that come about? Well, the Y chromosome has to come from the sperm. One X is going to come from the egg, but maybe you have an egg that got two X's, or maybe you have a sperm that got an X and a Y. That individual then is going to have certain characteristics when they have that syndrome, but they can survive. There's one XYY males. That one you can figure out. There's only going to be one way that can occur for an individual to have two copies of the Y chromosome. It's going to be a sperm produced through non-disjunction that has two copies of the Y chromosome. When it fertilizes the egg, now it's XYY. What happens when you have the extra Y? This one's pretty controversial. In most cases, it goes undetected. People don't even realize that they have an XYY genotype. But when people have studied it, they've seen that it makes males who carry it taller on average, and they have a few other characteristics. We also have XXX females, triple X, or sometimes even more than three X's. And there's some issues here as well. But the point is, if there are not too many genes on a chromosome, having an extra one or not having one, such as the Y chromosome, which has very few genes on it, isn't going to be so disastrous for the development of the organism that it can't survive. So all of these are only one selection of issues that go on from non-disjunction with sex chromosomes. There are more, but these are by far the most common ones. There we have it, non-disjunction and all the havoc it can wreak.